Hi, this is Andrew Olson from Dorian Ware's lab in Cold Spring Harbor, New York. I'll be giving the SorghumBase webinar today, just to make sure everyone knows this webinar is being recorded and it'll be made available online for anyone that couldn't make it, or if you want to refer back to it in the future. So the SorghumBase web portal went public a few weeks ago, and today I'll be showing you some of the genomic resources on the site. There we go. Nope. There. We're currently hosting the genomes and gene models for 5 sorghum reference genomes. Freshly sequenced and annotated genomes.
Andrew, I can't hear you.
Oh, come on.
It might be me.
Is it? I'm not muted.
He sounds fine to me. Maybe
okay. I'll just make sure I talk more directly into the microphone. Sorry about that. Let me make this go backwards. There we go. Yeah, so we have these 5 new reference genomes. Some of them are not so new, like the reference BTX623 has been around for quite a while. But generally, when you have new genome annotations, they don't have a lot of functional annotation on them. So we rely on comparative genomics to better understand these gene models. By building gene family trees, which reconstruct the evolutionary history of a set of related genes, we can bring in functional information from other crops and model organisms. So in the case of SorghumBase, we're including Arabidopsis, rice, maize, grapevine, and some other more distantly related species to cover the plant tree of life. We also have whole genome alignments with rice, which provide additional support for understanding sorghum genomic architecture. And we include the variation and expression data that has already been annotated on BTX623, as well as pathway data that was mapped to BTX623. These metabolic pathways are curated by the plant reactome on Japonica rice, and then projected to the other genomes using orthology relationships established by the gene tree analysis. Sorry about that. This diagram illustrates how the sausage is made, or maybe I should say sorghum brownies. We're using WordPress for our content management system. Think of blog posts, events, publications, etc. And we hope the community will come to use this system more and more to share newsworthy findings. In fact, several people have contributed blog posts in the past, but as of right now, with the site just going live, we're only showing the posts that are relevant to those 5 assembled genomes currently hosting. The front end of the site is powered by a Python Flask server. This application receives content from WordPress via a RESTful API. That's an application programming interface. And then it basically just serves web pages. Some of these pages are simple, others are rather complex with dynamic interfaces. For example, the genes page includes JavaScript code for a full web application that fetches data from various local and remote web services and lays out and renders the various components on the page. The genomic data that we're focusing on is loaded and managed in Ensemble databases. These databases are, and the associated analysis tools and web interfaces have been developed and maintained at the EBI, that's the European Bioinformatics Institute, for going on over 20 years. So this is a really robust platform, and SorghumBase will benefit from any upcoming developments to the Ensemble code base, which in some respects is showing its age. For example, we expect to deploy a completely rewritten genome browser when it becomes available sometime in the next 2 years, depending on their timetable. Our instance of the genome browser is hosted at ensemble.sorghumbase.org. But other things like this genes page are powered by other stuff. So this gene search index builds on the ensemble infrastructure by integrating multiple data types into searchable documents. It was developed for the Grameen project going on maybe more than 5 years ago. And we're leveraging that technology to support this updated implementation of the search interface. The APIs that support this interface are all open source and accessible for developers outside of this group. We've prepared a quick guide that demonstrates how to access the genomic resources we're hosting. And this guide covers the gene search as well as several ways to explore genomic data in the Ensemble interface. We also have a YouTube channel where we'll be posting short videos that demonstrate features of the SorghumBase web portal. So if you'll indulge me, I would like to play a 4-minute demo of the genes search interface.
My name is Andrew, and I'm the lead developer for the SorghumBase web portal. This video focuses on the genes search interface at sorghumbase.org. You can initiate a search from any page on the website by clicking on the spyglass icon in the header or typing the /key. Then just start typing and select matching filters. For example, to search for disease resistance genes would be the ND-ARC interval domain and leucine rich repeat regions. The grasses, you can do that. The search results page is organized into 3 areas. The panel on the left side shows the status of the search and the set of filters that have been defined. And on the right side, at the top, is a visualization of the genomic locations of matching genes, a count associated with each genome. Below this is a paginated list of genes, where each gene has a set of tabs that can be expanded to explore more details. The location tab launches a lightweight gene browser showing the gene structure. Expression tab loads baseline gene expression from various studies indexed by the UBI expression atlas. There's a homology tab that I'll show you later, and a pathways tab for associated pathways in Grameen's plant reactome. If one of these features is unavailable for a specific gene, it's grayed out like this, and a list of cross-references to other databases. Another search for a specific gene model. When there's a single search result, the homology tab opens by default. The homology tab shows the evolution of the gene family. The tree is interactive and has 3 display modes. You can click on a node to expand it and enable its children. The alignment overview mode shows a visualization of each protein in the multiple sequence alignment. Regions annotated with integral domains are color-coded. You can mouse over, click on a domain to see what the domain is. The multiple sequence alignment mode lets you see segregated alleles in coding regions of these genomes. For example, in this area are small insertions and deletions along these sorting lines. Gene neighborhood view shows 10 genes on either side of the gene family member. Genes are color-coded by gene family. You can highlight all the genes in a family by clicking on one of the members. This lets you see that there are 2 to 3 local copies of this gene in sorghum. You can also update the search by clicking on one of these buttons. This gives another way to see that there are 2 members of this family in 2 sorghum lines and 3 members in the other 3 lines. Here, Thank you for your attention and we welcome your feedback.
All right, so let's get back to full screen somehow. There we go. So this and so much more to come is made by and for the sorghum research community. We've established working groups, hosted visiting scientists at Cold Spring Harbor Labs, of course, pre-pandemic, and enlisted help with testing the SorghumBase portal. We look forward to working closely with many of you to provide valuable resources to support your research. I also want to acknowledge our scientific advisors, Steve Kresovich, Cleve Franks, and Mitch Twinstra for their feedback and guidance. And of course, the hardworking members of the WARE Lab at Cold Spring Harbor Lab. Our focus for the next few months is to work on integrating additional genomes and genetic variation data. We want to bring new data into the system as soon as possible to support your research. So we welcome your feedback to help identifying these key data sets. And finally, there are a few different ways to contact us and keep up with the developments at SorghumBase. We'll broadcast information of general interest to our mailing list. You can contact the team by email or through a feedback form on the contact page of the website. Will be available for regular office hours to help you navigate the site or learn how to better serve your research interests through your specific examples that the way you would search the site. We'll also be gathering feedback on the usability of the site so we can ensure that future development resources are dedicated to supporting the community. So send us an email to reserve a time slot for office hours. We also have a Sorghum Collaborators Slack channel. Email us also if you're interested in joining that. And finally, the next webinar will be held sometime in September to be determined, and we'll highlight another aspect of the site. So that's all I have. Thanks for coming to the webinar, and we'll open it up for discussion. You can either just chime in and ask a question or post something in the chat, and we'll look at it that way.
Hi, Andrew. Hey, my thanks to the whole SorghumBase team. This is Jack Okamura from ARS, Office of National Programs. I really like what you've done. I like the layout. I like the tools that you've generated and some of the utilities. And maybe the message to the community is if they get in early, then more personalized attention. So early users with questions or research needs, they could work with SorghumBase. Now's the time to do it. So thank you. It looks very, very good. I appreciate all the hard work by the team.
Thank you.
Marcella, thank you.
Else have any questions? I know we've worked, Scott has been reaching out to us pretty regularly and
Okay.
have had some, and so I believe has Joe. We wanted to make this short and sweet because it's really up there and out there now. And so we'll be able to start interacting with the community more frequently than we have in the past with the site up.
Sure. Sure. No, this is the way it always starts. So, any feedback from Scott? So, any thoughts, Scott? What are your impressions so far?
I think it's great. My question after today is, how do we, in our publications, how do you want us to cite this, Doreen? Do you want to just list the web address, or
So that's a good
or is there a plan to put something out there in more of a traditional publication form?
So Scott, we're working with there's going to be a special issue on sorghum and planta. And so we already have a draft publication in place that's had a first
Excellent.
review. So we're hoping that that will be the first set of benchmarking for that. So thank you for asking that. And in the meantime, if anything's useful beforehand, we'll figure something out. And as Jack mentioned already, you know, as we're starting to move forward, people who are interested in working with us, we would really use those use cases as an opportunity to push items through. As Andrew said, what we are really focusing on this first 6 months is bringing in more genomes with the existing infrastructure that we have to build up the content, and then working with the community to improve usability. So if there are no more questions.
How about Joe? You know, Joe, any comments or thoughts at this time?
Not at the moment. I'm kind of playing around with the website here, just looking at all the different stuff that's available. I'm happy to see there's a sweet sorghum in there.
Good.
The Rio genome.
Yep.
We anticipate in the publication that Andrew had, we're currently working very closely with the Australians to bring in the genomes that were in their most recent publication that represent the genomes that are more diverse outgroups, the wild species. Some of them have the wild species that they've been using. So we're hoping that we'll be able to update more frequently as we go forward. And as mentioned before, any of the publications where we have infrastructure that can handle that capacity right now, we really want to target and bring in and populate as rapidly as possible.
I also want to say I appreciate the partnerships and the other teams and databases, you know, working together with you. So I think that's important as well. So thank you for that.
Our intention is to jumpstart the working groups in October, but we made a decision, given that this is field season for most people, that it would be a better time to push, you know, get this out there, allow people where they have time to start looking at things, improve the population of the data, and then reinvigorate the working groups that we had pre-COVID.
Any announcements on, let's say, Maze GDB or some of the other databases? Can we help that way?
We plan on working with Maze GDB over the next month on that. Okay. We made a decision that we would have a bit of a, I wouldn't quite call it a silent release, but we wanted to go gently at first, make sure everything's operating, and then do more of a bit of a splash. We actually hope that we can bring in those other genomes within the next quarter, so as rapidly the next 2 months, if all things work out.
Sounds good. I'm very happy and very proud of you all. This is great. Great start. Thank you.
Thank you, Jack.